Welcome everyone to the Apothecary Tales, the basics of pharmacology. Today we're going to talk about one of the basic concepts but a very important major branch of the pharmacology, that is the pharmacodynamics. And in this video we'll be particularly talking about one main section coming under the pharmacodynamics, that is the drug-receptor interactions. So there are different receptors available and they act through various mechanisms. So we'll be talking about that in this video. So let's just get started. So pharmacodynamics describes the actions of a drug on the body and the influence of the drug concentrations on the magnitude of the response. That is simple, that is what the drug is doing on the body or the different actions of a drug on the body. So that is basically dealt with the pharmacodynamics, and usually the drug concentrations basically is responsible, or the influence of the drug concentrations play a very important role in the magnitude of the response. For instance, if you try to increase the concentration of the drug, gradually the response increases, as well as if you try to decrease the concentration of the drug, the magnitude of the response thereby decreases. So we can say it is actually proportional in nature. And most drugs exert their effects, be it both beneficial as well as harmful, by interacting with the receptors, that is specialized target macromolecules that is present on the cell surface or within the cell. So the drug-receptor complex usually initiates certain alterations in the biochemical and or the molecular activity of a cell by a simple process known as a signal transduction process. So now if you observe, when a drug comes and acts on a particular receptor, it will form a drug-receptor complex, and that would further initiate certain alterations in the biochemical and or the molecular activity of a cell by a process called as signal transduction. So what is this signal transduction? So first, the drugs here is acting as a signals, and whereas the receptors are the ones that is trying to detect the signals, so thereby acting as the signal detectors. Receptors usually transduce their recognition of a bound agonist by initiating a series of reactions that ultimately result in a specific intracellular response. So you have to keep in mind, the agonist usually refers to a naturally occurring small molecule or a drug that will bind to a site on a receptor protein and activates it. And there are certain secondary messengers or the second messenger or the effector molecules that are part of the cascade of events that translates the agonist binding into a cellular response. Now when we talk about the drug-receptor complex, the cells have got many different types of receptors, and each of which is specific for a particular agonist and produces a unique response. That means these receptors have the property of specificity. For example, the cardiac cell membranes have got the beta receptors as well as the muscarinic receptors. The beta receptors will bind and respond to the epinephrine or norepinephrine, as well as the muscarinic receptors will specifically bind to the acetylcholine. And these different receptor populations will dynamically interact with each other to control the heart's vital functions. The magnitude of the response is usually proportional to the number of the drug-receptor complexes. That is, if more amount of drug-receptor complexes are present, that means the response, the magnitude of the response is high. This concept is actually closely related to the formation of complexes between the enzyme and substrate or the antigen and antibody. Now when we talk about the receptor states, receptors usually exist in at least two states, that is in the inactive and active, that are usually in the reversible equilibrium with one another, usually favoring the inactive state. So what happens when an agonist binds to a receptor? It causes the equilibrium to shift from the inactive to the active to produce a biologic effect. Antagonists usually occupy the receptor but do not increase the fraction of the active receptor and may stabilize the receptor in the inactive state. Whereas some drugs such as the partial agonists usually cause a similar shift in the equilibrium from the inactive to the active form, but the fraction of the active receptors is less than that caused by an agonist, but still more than that caused by an antagonist. The magnitude of the biological effect is directly related to the fraction of the active receptors. Agonists, antagonists, and partial agonists are examples of ligands or molecules that can bind to the activation site on the receptor. So next we're going to talk about the major receptor families. So this pharmacology usually defines a receptor to be any biologic molecule to which a drug will bind and produce a measurable response. These enzymes, nucleic acids, and structural proteins can act as receptors for drugs or endogenous agonists. However, the richest sources of therapeutically relevant pharmacologic receptors are proteins that transduce the extracellular signals into the intracellular responses. So what are the major families? It usually consists of 4 families. They are the ligand-gated ion channels, G-protein coupled receptors, enzyme-linked receptors, and intracellular receptors. And the type of receptor a ligand interacts usually depends upon the chemical nature of the ligand. That means if the hydrophilic ligands are present, it usually interacts with the receptors that are found on the cell surface. This is because we know that the cell consists of the lipid cell membrane. So a molecule to pass through the cell membrane, it should be lipophilic in nature. So therefore, the hydrophilic ligands cannot cross the cell membrane. Therefore, they will just interact with the receptors that is present on the cell surface. Whereas the hydrophobic ligands, that is usually the lipophilic in nature, enter the cells through the lipid bilayers of the cell membrane to interact with the receptors found inside the cells. Now, talking about the first receptor, that is the transmembrane ligand-gated ion channels. The extracellular portion of the ligand-gated ion channels usually contains the ligand binding site. So this site is responsible for regulating the shape of the pore through which the ions can flow across the cell membranes. The channel is usually closed until the receptor is activated by an agonist, which opens the channel briefly for a few milliseconds. Depending on the ion conducted through these channels, these receptors mediate diverse functions including the neurotransmission and cardiac or muscle contraction. For example, stimulation of the nicotinic receptor by acetylcholine results in sodium influx and potassium outflux, generating an action potential in a neuron or contraction in skeletal muscle. On the other hand, the agonist stimulation of the GABA receptor increases the chloride influx and hyperpolarization of the neurons occur. Now, voltage-gated ion channels may also possess the ligand-gated sites that can regulate the channel function. For example, the local anesthetics will bind to the voltage-gated sodium channel, inhibiting sodium influx and decreasing the neuronal conduction. That is about the transmembrane ligand-gated ion channels. Next, we're going to talk about the transmembrane GProtein coupled receptors. So in these receptors, the extracellular domain of this receptor usually contains the ligand binding area, and the intracellular domain usually interacts when activated with a G protein or effector molecule. Now, there are many kinds of G proteins, for example, the GS, GI, and GQ. But all are composed of mainly 3 protein subunits. So the alpha subunit, if you observe, will bind to the guanosine triphosphate, and the beta and gamma subunits usually anchor the G protein in the cell membrane. Binding of an agonist to the receptor increases the GTP binding to the alpha subunit, causing the dissociation of the alpha GTP complex from the beta gamma complex. Now, these 2 complexes can then interact with other cellular effectors, usually an enzyme, a protein, or an ion channel that are responsible for further actions within the cell. Now, these responses usually last several seconds to minutes. Sometimes the activated effectors produce second messengers that further activate other effectors in the cell, causing a signal cascade effect. The common effector that is activated by the GS and inhibited by the GI is the adenylyl cyclase, which produces the second messenger cyclic adenosine monophosphate, whereas a GQ activates the phospholipid C and thereby generates 2 other second messengers, that is inositol 1,4,5-triphosphate and diacylglycerol, that is DAG, and this DAG and CAMP is responsible for activating different protein kinases within the cell. leading to a myriad of physiological effects. So this IP3 regulates the intracellular free calcium concentrations as well as some protein kinases. So this is all about the G-protein coupled receptors. Next is the enzyme-linked receptors. So this family of receptors usually consists of a protein that may form dimers or multi-subunit complexes. So when they get activated, these receptors usually undergo a conformational change resulting in increased cytosolic enzyme activity depending on their structure and function. This response usually lasts on the order of minutes to hours, and the most common enzyme-linked receptors such as the epidermal growth factor or platelet-derived growth factor or the atrial natriuretic peptide insulin and others possess the tyrosine kinase activity as part of their structure. The activated receptor phosphorylates the tyrosine residues on itself and then other specific proteins. Phosphorylation can substantially modify the structure of the target protein, thereby acting as a molecular switch. For example, when the peptide hormone insulin binds to two of its receptor subunits, their intrinsic tyrosine kinase activity will cause the autophosphorylation of the receptor itself. In turn, the phosphorylated receptor thereby phosphorylates the other peptides or proteins that subsequently activate other important cellular signals. And this cascade of activations usually result in the multiplication of the initial signal, much like that with the G-protein coupled receptors. Next is the intracellular receptors. So this fourth family of receptors differs considerably from the other three in that the receptor is entirely intracellular and therefore the ligand must diffuse into the cell to interact with the receptor. So naturally, in order to diffuse the cell membrane, what should be the nature or the chemical characteristics of the ligand? It should be lipophilic or hydrophobic in nature. The primary targets of these ligand-receptor complexes are the transcription factors in the cell nucleus. Binding of the ligand with its receptor generally activates the receptor via dissociation from a variety of binding proteins. The activated ligand-receptor complex then translocates to the nucleus where it often dimerizes before binding to the transcription factors that regulate the gene expression. The activation or the inactivation of these factors causes the transcription of DNA into RNA and translation of RNA into an array of proteins. The time course of the activation as well as the response of these receptors is on the order of hours to days. For instance, the steroid hormones exert their action on target cells via the intracellular receptors. Other targets of the intracellular ligands are structural proteins, enzymes, RNA and ribosomes. For example, tubulin is a target of anti-neoplastic agents such as the paclitaxel. The enzyme dihydrofolate reductase is the target of antimicrobials such as the trimethoprim. And the 50th subunit of the bacterial ribosome is the target of macrolide antibiotics such as the erythromycin. So this is all about the major receptor families. Next we're going to talk about some characteristics of the signal transduction. So the main two salient features of the signal transduction is that the ability to amplify the small signals and the mechanisms to protect the cell from the excessive stimulation. So first we're going to talk about the signal amplification. A characteristic of the G-protein linked and enzyme linked receptor is that they can amplify the signal intensity and duration. For example, a single agonist receptor complex can interact with many G-proteins, thereby multiplying the original signal many-fold. Additionally, the activated G-proteins persist for a longer duration than does the original agonist receptor complex. For instance, the binding of the albuterol, for example, may only exist for a few milliseconds, but the subsequent activated G-proteins may last for hundreds of milliseconds. And further prolongation and amplification of the initial signal are mediated by the interaction between the G-proteins and their respective intracellular targets. And because of this amplification, only a fraction of the total receptors for a specific ligand may need to be occupied to elicit a maximal response. Systems that exhibit this behavior are said to have the spare receptors. So what are the spare receptors? Those that in which a small fraction of the receptors when get occupied will produce a maximal response. So the spare receptors are exhibited by the insulin receptors, where it is estimated that 99% of the receptors are spare. This constitutes an immense functional reserve that ensures that adequate amounts of glucose enter the cell. On the other hand, in the human heart, only about 5% to 10% of the total beta adrenoreceptors are spare. An important implication of this observation is that little functional reserve exists in the failing heart because most receptors must be occupied to obtain the maximal contractility. So this is one important salient feature regarding the signal transduction. The next one is the desensitization and downregulation of the receptors. So what happens when there is a repeated or continuous administration of an agonist or an antagonist? It may lead to changes in the responsiveness of the receptor. To prevent the potential damage to the cell, for example, the high concentrations of calcium initiating cell death, several mechanisms have evolved to protect a cell from the excessive stimulation. So when a receptor is exposed to repeated administration of an agonist, the receptor becomes desensitized, resulting in a diminished effect. This phenomenon is known as the tachyphylaxis. And this is due to either phosphorylation or a similar chemical event that renders receptors on the cell surface unresponsive to the ligand. In addition to this, the receptors may be downregulated such that they are internalized and sequestrated within the cell, unavailable for further agonist interaction. That means when more amount of or overstimulation of the receptors occurs, the internalization or the sequestration of the receptors from the surface to the cell within the cell occurs. So what happens? The amount of the receptors available for binding to the ligands are reduced. And these receptors may be recycled to the cell surface, restoring the sensitivity, or alternatively may be further processed and degraded, decreasing the number of receptors available. Some receptors, particularly the ion channels, require a finite time following stimulation before they can be activated again. During this recovery phase, unresponsive receptors are said to be refractory. Similarly, repeated exposure of a receptor to an antagonist may result in the upregulation of receptors, in which receptor reserves are inserted into the membrane, increasing the total number of receptors available. And the upregulation of receptors can make the cells more sensitive to the agonist and or more resistant to the effect of the antagonist. So that is all about the signal transduction and the major receptor family. So let's just summarize what we have learned in this video. So we have learned about what is pharmacodynamics, the drug-receptor complexes, the major families of receptors such as the ligand-gated ion channels, GProtein coupled receptors, enzyme-linked receptors, intracellular receptors. And some characteristics of signal transduction we have discussed, that is the signal amplification and desensitization or downregulation of the receptors. So if there's any doubt, comments or suggestions, please do mail in us. Thank you.